Hi everyone, this is chapter 15. This is the last of our chapters for the semester. And in this particular chapter, we're going to be talking about antimicrobials. So the first part of this is 15.1. We're going to talk about the discovery of penicillin, the first antibiotic that was used. We're going to talk about the difference between what we call broad spectrum versus narrow spectrum. And this is as it relates to antimicrobials. Primarily, we'll be talking about antibiotics, but we'll also talk about some of the other antimicrobials. We're also going to compare the two vocabulary words of bacteriostatic and bactericidal drugs, and also some of the different ways that these drugs can be made or developed. And we'll talk about those terms as well. The last part of this that we'll talk about is therapeutic index, selective toxicity, and drug half-life. We want to think about when we develop a drug, the things that we need it to satisfy. First of all, it has to work well against the microbe. But secondly, we need it to not be dangerous to the host. Since we're treating a microbe that is within a host, we need to make sure it does not hurt the host. We'll also talk about how these can be administered, how we can sometimes have drug interactions, and also contraindications that would play a role in the drug development. The antimicrobial drugs radically changed modern medicine, and really what we're talking about here is looking at antibacterials. So the term antimicrobial drugs are any kind of therapeutic compound that can kill microbes or actually inhibit their growth. So one of the two, and we can categorize them by the type of pathogen they target. Antibacterial drugs are those that treat bacterial infections, and in general, these are referred to as antibiotics. We also have antiviral drugs that target viral infections, antifungal drugs that target fungal infections, and antiparasitic drugs. These can work on protozoans and helminths. One of the things that I want you to think about as we start talking about this is the idea that because we're treating the microbe in a host, we want to think about which of these types of drugs might cause the most effects to the host or the least effects to the host. And part of that is thinking back to the idea of which ones are we most similar to or our cells play a role in versus the ones that we have big differences with. So before antimicrobial drugs, people routinely died from infections that today we think of as posing little threat. And keep in mind that these safe and effective antimicrobial drugs were not developed until the 20th century, and really it wasn't until around World War II that these were used in high amounts. So how were these discovered? Alexander Fleming is the one who discovered the first antibiotic. He was studying Staphylococcus aureus, a bacteria that you guys are familiar with, and he went away on vacation, leaving some of his plates out on the table. And when he came back to the lab table, what he found is that some of these were contaminated with mold. But what he recognized from this, as you can see over here, there's lots and lots of colonies, but near the mold, there's actually an area of no growth of the colonies of bacteria. And so what he was able to determine from this was probably this fungus here, this mold, was able to produce something that was killing the bacteria near it. And so he started to think about how we could use whatever it is that this mold was producing to treat infections with this particular bacteria. And this mold was actually called penicillium, which is why we now have penicillin. And so what he determined was the mold was excreting this compound that was inhibiting bacterial growth. He named it penicillin after the species of mold, and penicillin was actually mass produced in the 1940s, just in time to save thousands, if not hundreds of thousands of lives during World War II. So when we think of antibacterial drugs, we want to think about them as exhibiting a spectrum of activity. And what I mean by that is that if you have a broad spectrum drug, so broad spectrum drugs, these are generally effective against a large population of bacteria. So in this case, we would expect that they would work on both gram-negative and gram-positive bacteria. So let's stop and think about why that would be important. Why would we want a broad spectrum drug? I can think of a couple of different times when we would want to use a broad spectrum drug. One might be if you have somebody who's very sick, but we don't know what's causing the infection. We want to make sure that whether it's gram-negative or gram-positive, the drug we give them will work. The second reason why you might use a broad-spectrum drug is the idea that you may have more than one bacteria causing an infection. So you may have a secondary infection, and if one of them is gram-negative and one's gram-positive, we would need a broad-spectrum drug. So what's a drawback? What's a problem with broad-spectrum? We already know that antibiotics can have unintended consequences by working against our microbiota. If we use a broad-spectrum drug, we would expect to have more bacteria in our microbiota that are killed off. So we might think about narrow spectrum drugs, and these ones target a limited range of bacteria. Perhaps they only target gram-positive or they only target gram-negative. These are usually preferred because they present less disruption to a normal microbiota. But what do we need to know in order to do a narrow spectrum drug? Now, looking back to what I said about broad spectrum, if we know exactly what the bacteria is that's causing the infection, we could use a narrow spectrum because we wouldn't be wondering if we were going to kill it or not. And also, if it's a single bacteria that's causing the infection, narrow spectrum drugs would work well here. So, antibacterial drugs offer two different mechanisms of action. We can have what we call bacteriostatic, and these just prevent bacteria from growing, so stop the growth of them. And these tend to target protein synthesis and metabolic pathways, and basically you still have some of the bacteria in you, but they're not allowed to grow. They're not allowed to replicate, really. We can also have ones that we think of bactericidal, and these will actually kill the bacteria. Generally, these target the bacterial cell walls, right? If we get punctures of the cell walls or the cell membranes, it means that the bacteria is not going to survive it. Same with the nucleic acids. If we target the nucleic acids, there can be no protein synthesis or replication of the DNA. So why would we maybe want to do bacteriostatic rather than bactericidal? I often think about gram-negative bacteria. Remember, they have an endotoxin as part of their outer membrane. It's the LPS, or lipid A, of their outer membrane. If we were to do bactericidal, this would flood our system with lots of that toxin and could actually cause more problems than if we just stopped them from growing. So, when we talk about this, a drug's bactericidal or bacteriostatic properties can actually change, though, depending on some different things. Depending on the pathogen type, what the dose is of this antibacterial drug that we take, the length that we take that drug regimen, also the pathogen load, how many bacterial cells do we have, and the route of administration. Is this something we take orally or intravenously? Bactericidal drugs might seem more effective, right? Because we can think of them as killing. We do run into some problems. That also means they can kill our normal microbiota. And as I mentioned a moment ago, they can lead to a spike in bacterial endotoxins that can be released that are deadly to us. So we can also think of the drugs as being either natural synthetic or semi-synthetic. So antibiotics are naturally occurring antimicrobial compounds. So think of that penicillin that was being produced by a mold. We can do synthetic antimicrobials. That means that we manufacture an antibiotic perhaps through a chemical process. And it might be that we know what that naturally occurring one looks like, but we want to manufacture one that's slightly different. We can also have semi-synthetic antimicrobials, and this just means that we take a naturally occurring antibiotic and we chemically modify that. And we've done that a lot with penicillin, that we have newer generations of penicillin medications. So one of the things, though, that we want to really think about is drug safety. So we want to be able to kill or stop the growth of bacteria, but we don't want to do that at the detriment to the host. So when we look at medical interventions, we know that they all have a risk-benefit consideration we need to look at. And the thing that we look at in microbiology that's most important is what we call selective toxicity. And what I mean by that is that it is only toxic to the microbe and not to the host. So we look for something that is selectively toxic, meaning that it only selects for that microbe, not us. And an antimicrobial that is the best treatment would be for a dangerous infection that, if an antimicrobial is the best available treatment for a dangerous infection, we may allow some side effects that normally we would not think of as acceptable if it's a saving of the life. So the thing that we look at for this is what is called the therapeutic index. And what we're looking at is a ratio of the maximum amount that a host can tolerate, or a safe dose, to the minimum amount that we need to be effective, or the therapeutic dose. And so what we think of is that we really want what is called a high therapeutic index, meaning that the amount that can be tolerated is huge. There's a huge amount that we can tolerate, and that the amount that is effective is very tiny. That is what we consider a high therapeutic index. It's considered much safer than a narrow therapeutic index. And if we go back to this ratio, let's say that the maximum amount of a particular drug that could be tolerated is 10 milligrams. We are fine up to 10 milligrams of it, but the minimum effective dose of it is 0.1 grams. That means that we have a ratio of 10 that we think of as very high. But if we think of something that would be a low therapeutic index, we mean the maximum our body could tolerate is 1 milligram and the minimum effective dose is 1 milligram. That ratio is a 1-to-1. That is low, and it means that we are likely to have very bad side effects. So the therapeutic index, the higher it is, the better it is for the host. So we also think about drug safety. We have to think of toxicity considerations. The kidneys and the liver are organs in our body that metabolize and eliminate drugs from us, and they can be susceptible to certain drugs. So we look for ones that hopefully are not nephrotoxic or kidney toxic or hepatotoxic, liver toxic. Those are two that we really want to be careful about, that they are not toxic too. So a quick checking into this, I want you to be able to define the difference between a broad spectrum and narrow spectrum antibiotic, and what would be the disadvantage and advantage of each. Why might we want to use one over the other? Again, I also want you to explain why the ultimate goal for antimicrobials is selective toxicity. What does that mean? That's a really important part of this chapter. And lastly, we want to think about the therapeutic index of a drug. Do we want a high therapeutic index or a low therapeutic index, and what does that mean? Can you explain why that is? That's the end of chapter 15.1, and I will be getting into 15.2 next. Please let me know if you have any questions. Thanks.